Hello all, hope you're doing great. In this video, we want to talk about CO2 fixation in C4 plants. In previous videos, we have talked about it in C3 plants, which had two specific videos, photosynthesis 1 and 2. And also we have talked about CO2 fixation in CAM plants. So now this is the last step and talking about CO2 fixation in C4 plants. C4 plants are like sugarcane or millet. You can see that just like CAM plants, because of the environment that they grow in, they need to have some supports, they need to protect themselves, so they have some extra processes in addition to what all C3 plants do for carbon fixation. We have talked about stages that CAM plants use for fixing carbon dioxide, CO2. If you remember, it was in two phases, day and night, and there were some specific stages and processes that could only occur in night because only at night stomatas were open. For example, entering the CO2 only happened at night, and something only happened in day. This is the difference between C4 and CAM plants. The similarity between them is just that both of them do the fixation in two stages, in two actual steps. However, the difference is that in C4 plants, there is no limitation in day and night. Everything just happened in days. This is the limitation, that nothing happened at night. And there is no actually restriction for whatever that is happening in day, because all of them happens in day, because their stomata is open in day, unlike CAM plants, which their stomata were closed in day. Okay, this is the sun, just for reminding that this is day, then all of these processes are happening in day. You now know PEP. You have seen in CAM plants video. They are a 3-carbon compound and they start the fixation process of CO2. For example, here in C4 plants, PEP, just like CAM plants, with the help of PEP carboxylize, join to CO2. And so this is 1, 2, 3. Obviously, with 1 C from CO2, it will be 1, 2, 3, 4. And as you know, again, this 4-carbon acid is called oxaloacetate, and it will change to malate. I'll just right away after that. All the way that I just told you now is exactly like CAM plants. However, in CAM plants, we showed that the oxaloacetate will be divided into two parts. One is PEP and the other is CO2. But in C4 plants, our malate actually divided to CO2 just like before, and not PEP anymore. It actually produces pyruvate. I know that sounds familiar because you had pyruvate in glycolysis videos too. We have talked about it so much in that video. But just as a reminder, again, just like PEP, this is another 3-carbon compound. So in C4 plants, malate will be divided into CO2 and pyruvate. However, this separation does not happen in mesophyll, just unlike the process of joining CO2 to PEP. It happens in bonded sheath space of the plant, okay? So in mesophyll, what we have is joining PEP to CO2 by the help of PEP carboxylize, creating oxaloacetate, creating malate, all of the things that happen in mesophyll, okay? But then when it comes to bonded sheath cells, the separation process happens. And CO2 just join the Calvin to produce sugar, which is our goal in all this process. And pyruvate just again get back to mesophyll again to be converted to PEP, because we don't need pyruvate, we need PEP to start over the cycle. This process, the conversion of pyruvate to PEP needs energy, and ATP is the molecule that creates this energy. However, unlike all the processes that we need ATP before, the ATP changed to ADP, here the ATP changed to AMP, not ADP, okay? So pyruvate, by using the energy of ATP and by producing AMP, will be converted to PEP. And again, PEP starts over by joining the CO2, which comes from atmosphere. The CO2 which combines with PEP in mesophyll came from atmosphere. However, this CO2 is the result of separation of malate into CO2 and pyruvate. The second CO2, it hasn't came from atmosphere. It's just in the plant, okay? So this is all the process of C4 plants. The difference with CAM is all of them happen in day, nothing at night. And the similarity to CAM is the first process which happens in mesophyll, because we had again PEP, which the help of PEP carboxylize join to CO2 and creates a 4-carbon acid. The 4-carbon acid will change to 4-carbon malate. The malate is non-stable, so it will be separated into two things, CO2 and pyruvate. And here is actually the first difference that we see in the process, because in CAM plants, CO2 and PEP produce. However, here CO2 and pyruvate will be produced. CO2, just for reaching our goal, goes to Calvin cycle, and the pyruvate, by using the energy and by creating the AMP in the mesophyll, as you see the process happening here, in the mesophyll will create PEP again to start the process. This is what we have for C4. And just as a reminder, I know we have a separate video, but just for showing again, this is what happened in CAM. As you see, the malate has changed to PEP and CO2. The CO2 joins the Calvin cycle, and this will go and start over the process again. And in C4, you see that pyruvate will change to PEP, and CO2 just again goes to reaching our goal, which is creating sugar in Calvin cycle.